I feel like I voted them all in, except for, of course, number 16. This one was strange because it was the results of SIM and CSIA, but I could have sworn I think I'm getting mixed up. But I submitted this one to that one. And then
to the 16?
Yeah, even though it says 17, it's the results.
So if it's exercise 17, it goes on to 17.
The results.
It does because it's 17 goes on to 17. So exercise 17 completing the unknown results goes on to the exercise 17 completing the unknown results. It's not necessarily. So if you can figure out which one didn't get submitted, where like that lineup got messed up, just submit it to the one where there isn't. Like if you didn't submit to 17, like there's an opening for that one, right? So you're missing there. Something didn't get 16, just submit it on 17. As long as I have it, I have the documentation that you submitted it. That's what's important, not that it's necessarily in the right place.
Okay, because I have to look at that because we don't get the results until the next day, even though we're what currently.
Right. So the in lab upload 16 is saying that this lab 16, everything on lab 16's page goes to lab 16. Not that the results have to be from like
what from here.
Because obviously lab 16 has lab 15 results on it, but the in lab 16 worksheet goes in the in lab upload 16. But that worksheet is still on 16.
That was tripping me out. Okay, so
it's fine. You can just, whichever one wasn't the 16, just put it on 17. Again, it's more important that I have I have documentation that you submitted it at all, and not as important that it be on the right one. As long as it's there, you're accounted for. Yeah.
Yeah, because also with here, with the litmus, because we often get those results on the whole. So I was a bit confused for doing this one.
Right. So that one. Like part of why I had you guys write it separately on the worksheet in that lab. I don't like to waste paper by reprinting and printing things. So these were originally from my summer class, and their timeline is completely different from you guys's. So a week later for them was exercise 16. For you guys, it was like exercise 15 or something. So you wrote your results by hand on 14. I just didn't want to reprint all new handouts for everything and waste paper on that, which is why we use the dry erase sheets anyways. But yes, so you don't have to fill that out. We can say that you recorded it earlier. That's fine. Yes. Yeah, because you have it somewhere, and that is more for your reference than mine. Again, when you guys turn these sheets in, I'm not looking for correctness. I'm looking for that you participated in the lab.
And understand.
Kind of. So when you come to me with questions about it, then I will help you with understanding. If you just turn something in and it has to do with the lab, if it's not correct, I'd still give you points for participation. So, yeah. Yeah, it's okay. That's not a bad thing. It's a good thing. And believe me, that's something I relate to in case you haven't noticed. All right. All right, lab Cahoots. Okay, so your unknown project. Sorry. Yeah. Yes. Sorry. I'm so used to the touch screens anymore. Yeah, if you want to pull that one up for me, and then we'll take a look at what I meant by that one. Oh, so it's the unknown project, not your presentation. Was that on the right thing? Did I put the comment on the right thing?
You 100% did.
Listen, it is a rough time right now for all of us.
You know what? I don't even know what I was thinking.
It's okay. Yeah. Take a look at it again. If it doesn't make sense, then of course. You're okay. It's a lot. We were just talking about that at the beginning of the class about how this is that time of the semester, man.
I don't know what I thought.
It's okay. Okay. everybody sign in, and then we'll go over this stuff. I did the review yesterday. With those, well, the one where I walked through each test, I went through the stains, and I went through all the second part of the semester tests. So if you haven't looked at that, I walk through each one and like write my own notes for it. I've also posted those notes. The notes themselves might be hard to make out because it's all in like black pencil because I didn't want to switch too much. I didn't want to take too much time getting it nice and pretty. But if you're having any issue understanding those notes that I submitted, then the video should help clarify that. And I think I covered most of the topics on there. I wasn't referencing anything for that. I was just going from what I remember, what we need to know for that. So that was what that was. And only 2 people joined. So I didn't have like enough like input on things that you guys might have questions about for it. So if you do have questions about the lab material, please feel free to email me or ask questions during now. So that if you are blurry on something, we can clarify that. Yes, but if you look at the sheet, it says wet mount motility. So that's, you have to be clear on that as well. You still have plenty of time to edit that though, so it shouldn't be an issue. Yes, any edits you guys need to make, again, midnight on Wednesday, you can still get those points back as long as it wasn't left blank. Yeah, no problem. It's a lot. I tried to get all that done last night. I sat down to like upload the videos and the Kahoot quizzes. And I'm like, man, while this is uploading, I'm going to go ahead and grade these things. And it's like, if I'm going to grade them, and I need to grade them all. So at like 2 a.m., I was done submitting the comments on all of that. I am happy to help. It's a tough time for all of us, not just you guys. Just want you guys to know that I'm trying to stay on top of things that you guys can too. I don't want you guys to be like, where's my grade for this? Or you said we could submit edits and you didn't ever tell me what I needed to edit. So now everybody's got their stuff. Before we get started with this, I am applying right now. Everything's due in April. If I don't say it, I'll forget. But for a doctorate in education. So that is more administrative. It's deceiving. It seems like it's for educating, but it's really for like administrative stuff in education. So higher ed administration. So I'm getting a doctorate there. I'm starting in the summer. So if you guys come around in the summer or this time next year, you can ask me about how my classes are going. I'll still be teaching full time with all of that too. But I figure there's too much here at OCOKC that needs improvement, and that my efforts might be better placed there. Not that I can't teach micro, but this place needs a lot of help, and I think that I can help with that. So yeah, so I'm excited about it. Come check on me. Yeah. I have a lot to say about it. You guys get to hear about it sometimes, but believe me, there's a lot more you're not hearing about. All right, let's get going. Lots of pictures for this one. All right, so technically everything we've learned about is fair game, but I've told you we're not going to have Kirby Bauer, not DNA, not the eukaryotes, and not ELISA. We'll have 24 to 26 stations again. I am going to have to edit how we had the positioning of the questions last time because I now share my lab with another class. So I can't just have all the stations taken up at all the benches and everything. So we're going to have to deal with one of the benches being empty. So it might be less stations and more questions at the station, but no more than 3 at each station. Okay, and I'll try to make them all the easier ones so that we aren't having a minute to answer like 20 questions at one station and everybody getting trapped there. But yeah, it'll be 50 questions, multiple choice on Scantron again. So it's exactly the same setup as last time. It's just one of the benches is going to be open. All right. So let's get into the basics. Let's get a mind in the game. What is most commonly used to inoculate our cultures from our choices here? All right, so loop, obviously, this is an easy one. Good job, guys. In which quadrant of a streak plate would you expect to find the most single isolated colonies? We have an image for you to refer to. Okay, so that's going to be quadrant 4, where we have the most diluted sample, and therefore the most spread out cells. One cell leads to a colony, then you can pick that one colony to create a pure culture, right? That's the goal with isolation. We saw this question on the last test. It will be back, same figure and everything. So just FYI, that'll be another relatively easy one. Right, so for our culture basics, remember what aseptic technique is and why we do it. Remember our lab safety. You guys do need to have your lab coats for the final exam. Probably going to have 1 or 2 points of the lab exam be associated with having worn a lab coat. So just be you have it on and you get the point for it. Lab safe seems stupid, but I don't know if you guys have noticed, like, it seems always like people will have their lab coats and then it's like the lab exams. They're like, oh my gosh, I forgot, because everybody just gets so in a tizzy. So don't forget your lab coat. Even if you have to borrow one, you would lose that point. Provide your own, please. Types of media, this is referring to broths, slants, plates, as well as our butts, right? So know the difference between them, which ones are solid and which ones are liquid. And then the different ways to inoculate them. Obviously, the loop on the surface of our solids, the stabs into the butts, and then using the loop in the broths, just swish it to inoculate those. All right, let's go through the stains. This is taking it way back. What is the first step in the gram stain procedure? After heat fixing, which I'm aware that's a thing too. Which stain is the first one? Old stain step. Crystal violet. So you guys learned a thing. You didn't know this before going to class, at least now you know this. Crystal violet, again, those molecules get stuck in a peptidoglycan. We fix them in place with our mordant, which is iodine. We will then rinse with alcohol to remove any of the crystal violet that didn't stick from adding that mordant. And then we will use safranin to counterstain the decolorized cells. Next question. What color do properly stained gram-negative bacteria stain in the gram stain procedure? It's just a random picture, by the way, not relating necessarily to anything. I don't even know what it's up. All right, so red or pink, however you want to think of it, safranin color, right? So they'll stain red. There's red or purple are your options here. Purple, positive. And now we know, having especially seen it on the exam, the difference between the pinkish and the purple. The purple is freaking purple. So at least we can delineate that. What is the name of this staining procedure that is going to be pictured? So if we look at this picture, you can see little like bubbles as well as little greenish, bluish, teal colored parts. Some of them aren't bubbles, some of them are just parts in the actual red structures. The red structures are our vegetative cells that are actively multiplying. The little teal color guys are our endospores in the endospore stain. They look bluer in our microscope, but rest assured it's still an endospore stain. Which of the following bacteria will be acid-fast positive? This is a mixture of an actual acid-fast stain. The slide that I have for our acid-fast procedure looks quite a lot like this. It's a mixture as well. It's the only slide I have. You guys will see the same exact slides again. Might be different questions, but they are the same slide. Okay, so that's going to be Mycobacterium species. Remember Mycoplasma? Plas is always myco. For Mycobacterium, we're looking at the mycolic acid. That's that wax. The wax in the mycolic acid, that interacts with the lipid soluble carbofusion primary stain. It stays stuck to that wax, and we'll rinse off of regular negative cells that don't have mycolic acid. Mycoplasma, plasma, again, I always end up carrying an actual bucket of like plasma from someone's blood. If you spill it, it doesn't have a shape, just like mycoplasma bacteria don't have a shape because they don't have a cell wall. That's their defining characteristic. those, yes, I had to think about that one. After the slide has been heat fixed, when do we use heat again in the acid fast stain procedure? Okay, so this question is a little bit of a trick question, but it's also testing that you're actually reading and comprehending the question. So acid fast stains don't have heat at all. So the answer is no, because we don't use heat on this one, right? Do you guys remember which procedure does use heat? Endospore. So to actually fix the stain, endospore, right? That's that steam that we'll use for that one. It's going to be a question, by the way. What color is a negative acid fast stain? All right, so blue. Our counter stain for acid fast is methylene blue. Your primary was carbonyl fuchsia, which is fuchsia or pinkish red. Okay. What is the name of the primary stain used in the capsule staining procedure? It's one that gets a lot of people. This is an image of a capsule stain. If you look at this picture, is this organism, does it have a capsule? It does. So the reason we know it does have a capsule, if you don't have a capsule, you won't have any white on that image at all. So the white around those little pink dots, between the pink dots and the purple background, the white is the halo caused by the actual capsule coating. It doesn't stain. So Congo red is our primary stain for capsule staining. We do not heat fix. We mix it, we spread it out, we air dry it, and we added those drops of medieval stain. That's what makes the purple background. And then the halo means there's a capsule. If there's no capsule, it will just be pink, butted up right against the purple, no white. All right, in which staining procedure do we use steam? Hey. All right, this is the endospore stain. Where basically this is the one where we sat over the freaking boiling green thing, dropping green dye on everything for 10 minutes over that beaker. That forces the heat forces the green stain into the endospores themselves. Even though they look a little bluish in our microscope, again, it's a green stain that we're using called malachite green. If you remember that it's green and that we had the green and the red in the picture, hopefully that will help you remember the green thing was the steam thing. That should help too. We will have questions on the test, sort of like the acid fast. I asked if when we use steam in acid fast, right? It's a trick. We don't. You will have questions like that on the test where I will ask you, what is the pH indicator of this test? And it may not have one. You'll have a whole list of pH indicators that we actually use. And one of the choices might be we don't have one. You'll also be given tests that do have pH indicators. And I'll ask you what the pH indicator is, list of all the pH indicators. And one of the choices might be we don't use one. So you'll have to know which ones have pH indicators and what they are. And usually I like to include options of bromothymol blue and bromophenol blue. Which one is the correct one? Thymol. Okay, pay attention to your choices. All right, so for the stains, know your procedures, the steps, what each stain literally is for each step and why we're using it in the step, because they all have a purpose. Know how to identify the stains just by looking at a slide that I give you. You guys know this, it's the same slides. It's the same steps as in the midterm. Brush up on them. You know what you're gonna see. Now know them well enough that I can ask you anything about them. Differential and selective media. What type of media is pictured? This is another question that is pretty common on the test. A lot of the test questions are like this. I'm gonna just give you a plate and ask you what media is it? All right, so this one is EMB. Good job. This is EMB, and we know that it's EMB. Number one, it has that darker color to the auger itself. Number two, the dead giveaway, that metallic green growth. Only EMB can have that. None of the others do that. All right, so about EMB, what is the selective agent in EMB? It's selective and it's differential, but what's the selective part? All right, so our selective agent for EMB is eosin methylene blue. Don't beat yourself up too hard on this one if you pick biosalts, because EMB and the one that has biosalts are pretty similar in their actual differential part. So they both test for lactose use, and they both can have pink growth associated with lactose use, right? So I understand that confusion. Just be sure that you're aware that eosin methylene blue, the EMB, eosin methylene blue, is the selective agent for that one. The other one, McConkey agar, right? It has biosalts for the selective agent. So you're not wrong. You're in the same realm. Both of them test for gram negatives too. Alrighty. Which media is pictured here? Some of what we just discussed should help give away this one. Right, so that's McConkey agar, and we knew that because of that pink growth on it, and because the media itself was not that dark color, really. That's what we know for identifying this one here. If I feel like there is a vagueness there where I feel like media looks very, very similar to one another. For example, when we looked at McConkey agar uninoculated and blood agar uninoculated, they look very similar. I'm not going to place one in front of you and be like, which is it, and give you those two choices. I'm more likely that this is McConkey agar. Now answer some questions about it. So I'll try to keep that vagueness out of the test wherever I'm able to perceive it. If you take a test, if you answer the questions on the test and you think you've met one of those vaguenesses, then you could always ask. Okay? There's no harm in asking at any point. Worst case scenario, my answer will be, well, we talked about it. Is this media selective or differential? So this is McConkey. We've established that. Is it selective or differential? And you can select more than one. All right, great job, guys. It is both selective and differential. Selective with those bile salts and differential for lactose use, whether it's pink or not. And also whether it grows or not also tells you something on that selective side for that identification purposes. All right, which media is this one? All right, so this is mannitol salts agar. It's pretty identifiable. You have that classic coral red orange as our neutral. We have the yellow on the acid, and we have the pink for our alkaline, right? So that's phenol red, in case you haven't caught on to that. So that's our pH indicator here. So that's our pH indicator. That's the differential side. Our selective side for McConkey agar is salt. So gram-positive organisms with their thick peptidoglycan wall are going to do better with salt content because they aren't susceptible to plasmolysis, or that shrinking in and death of the cell from osmolytic pressure. Right. What is the pH indicator? We only have 3 pH indicators altogether. So the first one we very, very first got interested in and got introduced to, is what I was trying to say, was phenol red, and it was with mannitol salts agar. And so we've seen it repeatedly since then. But phenol red, and here we will have our acid. We'll start with, yeah, let's do acid. That's fine. Acid, neutral, and alkaline. So for now, we're talking about phenol red. Acid is yellow. Neutral is like orange red. It's coral colored. And then we have alkaline is pink. And it's a pretty vivid pink, right? So we'll have two others that we're going to get review over today as well. And one of them relates to a type of media that has a lot of different possible answers that are all going to be a lot more clear if we can put them on a little chart I'm going to create for you guys. All right, so differential and selective media. Know what selective means, know what differential means, know what the selective aspects of a media is, and what its differential aspects are. Any color changes, is it the actual growth changing color or the media changing color? pH indicators really on our differential and selective plates. We just had phenol red, and you really only need to know about it in mannitol salt agar, okay? Staph, we didn't talk about the staph media here, but I'll remind you guys that Staphylococcus doesn't just mean Staphylococcus aureus, right? There are other kinds of Staphylococcus bacteria like Staphylococcus epidermidis that we learned about in chapter 19 as a major contributor to your biome. So Staphylococcus media is favoring all Staphylococcus, and that one is only selective. It doesn't tell you any differences between them. It's whether you grow or not, and it's got a lot of salt in it for that purpose. But mannitol salt, because we have the salt there for that separation of the selective side and the differential of the phenol red, now we can see if we can use the mannitol, right? And if you can use mannitol, you make the acid, you turn it yellow. Staph aureus does that, whereas Staph epidermidis doesn't. And I won't necessarily test you on that, but you can see if I'm using just staph media, it's not really going to give you an answer if it's Staph aureus or something other staph, right? But mannitol salts can be used for that. It has more to it. So if you're trying to identify your staph species, you might use other tests if you're using staph media, or you might just use mannitol salts agar. It just depends. But remember, there's not just one staph out there. Blood agar, keep in mind the 3 different kinds of hemolysis. One of the things that I had a problem with on that lab, not for everybody, but one of the things a lot of people were getting wrong in the lab was they would say that they got growth on the blood agar, but they didn't say what kind of hemolysis was there. That's part of our identifying process. If you grew on it, you need to be saying what kind of hemolysis it was. If you didn't see hemolysis, guess what that means? Gamma hemolysis. That was the answer for that. Beta is our clearing and alpha is partial. So you have green from partial breakdown of red blood cells. The absolute most missed thing on the lab report by far, and that a lot of you guys got comments on, is that we did not ever test for capsules for our gram positives. And I had answers from almost every single student for the gram positive for the capsule. Not sure where we got that from. Second was the motility. So the motility in the top part is referring to what we saw on our actual live microscope slides with the wet mounts. There's another motility for the SIM. Those are separate. They should align, but if we didn't do the motility wet mount, you shouldn't have data there for your gram positive. So that was another thing. So that's part of just being aware of how to fill out the forms correctly and what the difference is in those tests as well. All right, biochemical tests. Let's go to our tubes and some plates too. Which test is this one in the picture? You were just telling me I just now stood up. You gotta be kidding me. Whatever. All right, so this is the citrate test. Again, a very common question on the test. They seem simple enough, but we've got a lot of tubes to recognize. So I'll literally like a green tube and line that as a blue one and ask you, what test is it? And that might be a question, right? So this is citrate. We know it because it's green and then there's a blue and it's a slant, which you might not necessarily be able to tell from the picture, but all the tubes you can actually handle in the lab. There will be their physical tubes. You can pick them up if you need to, and that. So it'll be stuff you've actually worked with and will be familiar with. All right, which of those 2 tubes was positive for the test? This is my fault. I need to get out of here. because this is their time. So we'll quit here and move into 101 if that's okay with you guys, because I don't want to take up their class space. That was my fault. Okay, so the tube that was positive was B, blue. We'll talk about bromothymol blue next. And get out of here, get out of their hair. Jeez, man, I thought I was so prepared. I was already recording it. We'll rejoin on Zoom if you guys are on Zoom.